that. Yeah, welcome everybody to the Coffee Chats. My name is Dennis Koester. I'm the Director of Industry Research and Relations here at Drug Hunter and your host of this Flash Talk, or Coffee Chat, I should say. I'm here with Hesse and Lew Pennington. Hi, how are you guys doing this morning?
Great. Well, thanks for having me along, Dennis. It's always a pleasure to
Wonderful,
talk
Dennis.
to you.
Thanks.
Yeah.
By the way, what is that on your tie?
Oh, that's an antibody. I put like a scientific touch to my bow tie today. So yeah, just wanted to spice it up a little bit, right? Yeah, and for the participants here, when you're dialing in, you can, again, you can see and hear us, but unfortunately we can't see and hear you. Maybe there's a format in the future where we will enable that. But please to interact with us, please, the chat function, and people already mentioned where they are dialing in from. So we have people from the Netherlands. It's 6 p.m. in the Netherlands. So welcome to you guys. Happy good evening, Friday evening. That's amazing. We have Yoshi dialing in from San Diego. So 9 a.m. in San Diego. Good morning, San Diego. A lot of more people from San Diego. We also have people from Maryland. Later on, please, if you have any questions, we're going to have a brief presentation here. Please use the Q&A feature, which is distinct from the chat box, to interact with us. And of course, as you're used to, you will be able to find this recording on YouTube in about 48 hours. And we also will have the presentation that we'll share today on the website available as well. So why don't we dive right in? Today, we're going to be talking about the patent highlights. So I did want to give a brief introduction to what Drug Hunter is for everybody of you who may not be familiar with Drug Hunter. And, then we'll launch right into the patent highlights, and I'll show a little bit of how we put this presentation together and what we do with the patent highlights. And Matt, maybe during this presentation, can actually speak a little bit more to that, because Matt is our editor for the patent highlights. So yeah, Drug Hunter is actually drug discovery distilled. So as you know, companies disclose discovery strategies and tactics like every day, and the knowledge is really fragmented across different sources. So may it be like in patents or publications or conferences that you maybe not have the time to go to. And Drug Hunter really distills this knowledge together and makes these findings from other company accessible to you, so that you can basically, you don't have to reinvent the wheel. You can essentially learn the lessons that other companies have learned the hard way and apply those strategies to your own discovery projects. So to do that, we go through a variety of different sources and bring this together really in a single searchable knowledge base, filtering out the noise for you and really directly getting to the signal, to the information that is relevant for you. So we're focusing on things like strategies and tactics, where we talk about target rationale, but we really in our case studies cover the discovery story end to end, where we talk about the hit finding strategy, any problems that occurred maybe during the lead optimization, also clinical differentiation, before that, how the clinical candidate was nominated, and we finish up with the synthesis as well. And we do this for a variety of different modalities as well. We talk about the technologies that were invented on the way, and everything is really searchable by structure, but also by keyword, so that you can easily find articles where you just have to remember one part of the story. Oftentimes it happens to me that I forget the entire story, but maybe I remember one part, a specific toxicity or something like that, and then you can find that story by using this particular keyword. Or you maybe don't have the entire structure, but you remember there was this particular structural motive in the compound, so you'd be able to use the structure search for that as well. Or to design compounds, you would be able to use the structure search as well to see if this compound maybe has been in clinical candidates or maybe even in an FDA-approved drug. Drug Hunter is trusted by hundreds of organizations at this point that are Drug Hunter members. So we serve a broad client base from large pharma companies such as Pfizer, Takeda, and Amgen, but also biotechs that like discover really innovative new technologies. And we also have some academic subscribers as well. So really encourage you to check out the website. And actually, I think what differentiates Drug Hunter the most, and you'll see that today, is actually our fantastic team. So we assembled a team of really superstar scientists. Lew Pennington here on the call as well with us, our scientific director, with more than 30 years of experience. Then Matt. We've both been at Novartis for the better part of 7 years. Matt, after Novartis, took a director position at Sage, looking more at depression, so really challenging disease area. Rory's been, before he joined us, a medicinal chemist at Insight, looking more at oncology drug discovery projects. And of course, our founder and CEO, Dennis, has also seen both the bigger pharma industry as well as the biotech and how they operate in different modalities and with different targets as well. We've been able to really assemble a superstar scientific advisory board headed up by Wendy Young, our chairwoman, but also in the earlier, Nick Meanwell, Dean Brown, Dan Erlandson, and Ken Bremold joined this scientific advisory board as well. And with that little intro, I want to show, we're going to talk about some patent highlights today. We've picked out some nice targets for you to, really do a deep dive on. But before I go into that, I briefly want to really show you how we assembled this on Drug Hunter, how we assembled this presentation. So for that, I'll just briefly share the website. So hopefully you can kind of see my screen here. To find the patent highlights, you can, or the patent search, you can just go to resources here on that tab and then go to the patent search, which is located here. And then you have that database available to you where you can just type in an interesting target. So let's try, GLP-1, for example, or we can try a PTPN. Those are some interesting targets that we'll cover today. And what this allows you to do, you can also just type in a disease area and talk about find out what kind of targets are worked on in this particular disease area. But if you're looking for a particular target, what you can do is, without you opening the patents, on the right-hand side here, you see a high-level summary of what can be found in the patent. So may that be any the number of compounds that's been patented, that's often an indicator for how important the patent is. Any biochemical or cell data that is disclosed in the patent. Oftentimes we'll also talk about PK or efficacy data if that was disclosed. Really a jump starter for any patent research you're doing. And then we're also going one step further here. If you just go here to that full text search and type in patent, what you can do is you get to our patent highlights here, and this purple tab here is also functional, by the way, so that you can get your own roundup of what's been happening in the patent space. But every month we kind of distill the top 6 to 10 articles, top 6 to 10 patents down, and a lot of the content that you'll see from today is actually taken from these patent highlights. And with that, let's launch into the science. And there I'll show briefly the agenda. So for the agenda, we will start with, let me go through this real quick. So we will start here with what I just typed in there, PTPN 1 and 2, and then SARM 1. I'll hand it off to Matt and Lew, and then we'll finish with GLP-1 and VAF1. So those are the targets we're going to be talking about today. So PTPN1, really interesting target. And the reason I selected this as one of my favorite patents, actually, is I saw this acquisition by Boehringer Ingelheim. They acquired this company based in San Diego called Neuro for a $1.3 billion payment, or up to $1.3 billion payment. And this is for a preclinical program. So naturally, I got really curious about what this target is all about. This is a checkpoint inhibitor. And yeah, I was really curious to see, what is this target all about, and what are some compounds. So I used Drug Hunter's patent search tool to dive into the literature here. So it turns out PTPN is a really interesting negative regulator of the insulin and leptin signaling pathways. So it could actually be an interesting target for, type 2 diabetes or obesity and add to the really hotly pursued targets like GLP-1 that the GLP-1 receptor that we'll be talking about as well. But also PTPN is involved with the cancer progression, particularly for breast and lung cancer. And, it's really involved in regulating immune signaling and, has its inhibition may actually show and enhance an anti-tumor response. So this is actually a phosphatase. So phosphatases are traditionally really difficult to drug, as you can see, for example, with SHP2. They typically have highly conserved binding sites, and the binding sites are very polar. So that's why it's really difficult to drug. So, diving into the patent literature with Drug Hunter's tool, I was able to pull out some really interesting chemical structures. And what I found here in the Nerio patents was they all have this really interesting common motive is the thiadiazolidinone dioxide here, this type of ring, and they're all flanked by this ortho-substituted, orthofluoro-ortho hydroxy substituted arene. And, those structures are all really the same. And diving into other patents that we just saw from, for example, BMS and in silico medicines in the space, they also all feature that, really recognizing motive. So I really wonder now, what is the differentiating factor that Nerio's patents have? So this is something that really remains to be seen. But, it's a super interesting target to watch. And we have chemical matter on this particular target that shows sub-micromolar activity here, even in cell-based assays. But I believe this is really, with that really hot acquisition, this is certainly a target you want to watch. As the next target that I want to cover is SARM1. SARM1 is really an enzyme that's enriched in neurons and is activated in response to neuronal injury. So actually, inhibitors can be used probably for the treatment of neurodegenerative diseases such as, ALS or Lou Gehrig's disease, potentially even MS or like peripheral neuropathies. And, the interesting thing in this space was, Disarm Therapeutics was, which really has an apt name here, was acquired in 2020 by Eli Lilly for this particular asset that they have here. And that's a covalent, interestingly, a covalent, somewhat unusual warhead, if you will, for a covalent SARM inhibitor. But they really interestingly found this out by doing washout assays and still seeing activity after the compound was washed out. So Lilly really entered this space here in 2020, but Neura Bio, which actually just announced in September, on September 17th, additional Series A funding, is somewhat the leader in the space. And, you can really actually use these patent highlights to predict what compounds are going to become hot. We actually reported on those patent highlights in July. And, in September, this acquisition happened. So really, really a way for you to kind of track interesting hot chemical matter. So coming back to Neura Bio really has, an asset in phase 1 clinical trials that they brought into clinical trials in August last year. And the chemical matter is undisclosed, but it's likely to be related to this compound here, NB7. So, really has a different MOA, of course, to these covalent ones. Really interesting to kind of watch that space. Another company who's been really active in this space is Denali Therapeutics. And as you may have heard, Denali Therapeutics is actually spinning out their small molecule research into a company that we believe is currently still in stealth mode. But they've also been really interested in this particular target, SARM1. So they've published patent application with this urea-based chemical matter here, exemplified by this example 84. So, that is also certainly a non-covalent mechanism. They've, published on these types of inhibitors in late 2023. And also in 2024, they've had a paper that, detailed the biology of SARM and how SARM1 is involved in, neuronal, the biology, the details of the biology, how SARM1 is involved in neuronal degeneration. So in June, actually, this year, they disclosed another series that's a non-urea series that really showed, really high potency and is very much differentiated from the earlier chemical series that they've been patenting. AstraZeneca, what you can tell, like here on the bottom right-hand side, and they were able to show not only that it had some biochemical activities, but also that they had some that they were able to kind of protect rat dorsal ganglia from a degeneration at sub 100 nanomolar concentration. So really interesting space here to watch, a really hot target to follow for a very challenging disease indication. And with that, I'll hand it over to Matt to talk a little bit more about CARS inhibitors. Matt, stage is yours.
Yeah, no, one of the questions we get asked a lot is how do we select these patents, which we highlight each month. Some of it is, we're looking for hot targets, we're looking for trends as, different companies move into different target areas, different disease areas. One of the other things we like to look out for is what are the novel targets? What's something that we don't recognize? And actually, CARS was something that jumped out to us because it wasn't something that I was aware of before. And while this patent is based on CARS inhibitors, it's actually really a NRF2 KEAP1 story. And obviously, the NRF2 KEAP1 axis has been really hot lately. There's been a lot of kind of disclosures in it. Last year, Reata and Biogen's omethoxazolone was approved for Friedreich's ataxia. And NRF2 is this transcription factor, which really is key in a lot of cellular processes. But one of the things in terms of therapeutic importance is it promotes the expression of anti-inflammatory and antioxidant genes. So if you can change that, either kind of upregulate that or downregulate that, different indications that might be interested for. So obviously, if you downregulate NRF2 expression, which is how KEAP1 actually engaged with it. So KEAP1 is an adapter for an E3 ligase, which degrades NRF2. So if you inhibit KEAP1 and upregulate NRF2, you end up with increased expression of these anti-inflammatory and antioxidant genes, which would be really important for, say, chronic inflammation indications. So looking beyond Friedreich's ataxia for chronic kidney disease, neurodegeneration, aging. But actually, there's been more and more people starting to look at now, how do we inhibit this degradation of NRF2? How do we increase this degradation of NRF2 by activating KEAP1? And actually, we had a nice story from Vividian, had their first-time disclosure at the ACS, and we had a nice coverage of VDD130037, which is the first covalent activator of KEAP1, which is looking now at oncology, because in a lot of there's certain cancers whereby NRF2 is upregulated, and that's actually driving the cancer. And this is actually where this patent comes in that CARS is actually a really important part of the protein synthesis machinery of cells. So it's a lysyl tRNA synthetase, which is obviously key in charging tRNA for protein synthesis. And you can imagine these might be pretty cytotoxic. And so what Novartis has done is actually gone with a prodrug strategy here. So they have on the left their drug prodrug as a ketone here. And what they've done is they found that in NRF2 overexpressing cells, you get a increased expression of this arrow ketoreductase, AKR1C3. And what this does is it selectively reduces that ketone down to the active alcohol. And this only happens in cells which have this NRF2 upregulation. So you have selective activation of the drug in cells which are tumor cells which are driven by NRF2 overexpression. So it's a really neat little trick here. And you can see that on the left-hand side, you got this HCARS AMP assay. We have greater than 10 micromolar, but when you have it as the alcohol form, you now have 9 nanomolar activity. We've reproduced from the patents here down at the bottom. You can see 2 cell lines. So on the left, you have dose response in a KEAP1 mutant. So if you have a KEAP1 mutant where it's now inactive, you have increased levels of NRF2. In this cell, you can see that you have a really, really strong effects where, at the top 2 doses, 150 and 200 mg per kg, you have like complete tumor regression. And then on the right-hand side, you have another mutant cell, which only has middling expression of this keto reductase. And here you can see much more moderate effects of the drug. So I think this is a really neat use of this. And actually looking further into CARs, in August, there was another patent which we covered from a small South Korean firm called Zymedi, where actually they just got a orphan drug designation from the FDA for their monoclonal antibody for against cell surface expressed CARs, and this is for treating pulmonary arterial hypertension. And they actually had a small molecule set of inhibitors as well, which they published in August. So it's, like I said, we're always looking for these kind of these different trends and these different themes, and who's coming into this area that you might not expect. So if we go to the next slide. I also wanted to kind of talk a little bit about DICE alphas, which is a kind of fully owned subsidiary of DICE Therapeutics. They're small molecule IL-17A inhibitors. And obviously, IL-17A has been a big market for a long time here now for psoriasis, ankylosing spondylitis, and psoriatic arthritis. And obviously, this is a kind of a nice story in terms of you've got these big important cytokines, and obviously these are not the most druggable proteins, and so typically they've been engaged with these monoclonal antibodies. Novartis, secukinumab, Cosentyx, which made $5 billion last year. You obviously want to think of these as being targets of monoclonal antibodies, but then you actually end up moving to kind of, can we engage these with a small molecule? And you can see here that the first couple of disclosures around here were based on these larger macrocyclic peptides here. There's a nice kind of analogy here with thinking about, say, PCSK9, right, where you have the monoclonal antibodies, but then Merck comes on and says, actually, we have this macrocyclic peptide from mRNA display that we can engage it. The next step from that was obviously AstraZeneca's disclosure at the ACS, which we also disclosed and covered of their AZ0870, which is really a genuine small molecule inhibitor. So it's nice to kind of like see you go from these big biological systems to macrocyclic peptides to actually you can drug these proteins with small molecule inhibitors. And so covering these IL-17A inhibitors, obviously Ensemble were one of the first, and they actually shut down in 2017, and their IL-17A programs picked up by Novartis, makers of secukinumab. There's not really been much about that anymore. Really, the leaders had been Leo Pharma, and they are still actually interested in this pathway. Their topical JAK inhibitor cream, delgocitinib, was just got an NDA accepted at the end of September for topical use. But actually, their IL-17A is no longer in their pipeline, so I think they're kind of moving kind of downstream towards JAK inhibitors. DICE Therapeutics, though, was picked up in last year by Eli Lilly, which is really making big moves in the immunology space. They picked up DICE for $2.4 billion. We actually have some nice coverage of that in our 2023 M&A coverage series, which came out earlier this year. And what they were looking for really was their two clinical candidates, DC-806, which is in phase 2, and DC-853, which is their own fast follower program, which is in phase 1. Like I said, Eli Lilly has been making big moves in this area. They just a couple of months ago picked up Morphic for $3.2 billion for their integrin inhibitors. So it looks like this IL-17 space is actually going to be really interesting. These compounds here, which we covered from these two patents, you can think that they might have a similar type of binding site to the Leo Pharma molecules. We could draw these more suggestively. But I think this is one of those things that we really want to keep an eye on what is coming up from DICE Therapeutics and see if we can match it to actually what's going on in terms of the compounds in the clinic. Some of the other patents which came out recently in the IL-17A space that we've covered in our patent roundups, so UCB Biopharma and Sanofi had stuff earlier this year. And actually what really interesting that came out in August was Biohaven Therapeutics had an IL-17A LITAC degrader, which uses the lysosomal, which kind of drags it into the cell and degrades it that way. So I think IL-17 small molecule space is really one to keep an eye on.
Yeah.
Right, great.
With that, I think I'll pass over to Lew. Sorry.
All right. Oh, no worries, Matt. Thanks. So yeah, as Matt mentioned at the beginning of his presentation, there's various factors we look into when we're selecting targets. So for the first target or first patent application I'll be highlighting today, this is a new target in an area that's actually becoming quite hot. So I'm going to be talking about Accent Therapeutics DHX9 inhibitors for targeted cancer therapy, and I think you'll see in a little bit why this is a pretty cool target in a area that's pretty hot. This is the most recent patent application from Accent Therapeutics. They published at least one other application that I'm aware of. And to our knowledge, they're the only company that's disclosed a DHX9 program. So this target to us is quite novel. So what is DHX9? It's an RNA helicase, and it's involved in a whole host of things. It's involved in transcription, translation, splicing, editing, and even genomic stability. And it binds and unwinds various nucleic acid structures, including double-stranded DNA and RNA, single-stranded DNA and RNA, DNA hybrids, circular RNA, and even G quadruplexes. So this is a protein that does a lot of work in the cell. So DHX9 is also believed to be a significant factor in cancers with microsatellite instability. And that's probably the key word for the hot area that I'm talking about. And also cancers that have defective mismatch repair. So as a lot of drug hunter members will know, perhaps, microsatellite instability high cancers have really come into the spotlight recently due to the flurry of activity around Werner helicase. So we've covered this helicase in substantial detail at Drug Hunter, including highlighting the Novartis's HRO 761 and Vividian's VVD 214. So if you look at figure 4, you can see that these DHX9 inhibitors show greater potency in an MSI high colorectal cancer cell line than the microsatellite stable cancer cell line. And if you look at figure 5b, you can see that compound 18 exhibits similar microsatellite instability sensitivity across cancer cell lines. And this is exciting because this means in these types of cancer cells, it could have potentially fairly broad applicability. And then finally, and probably most excitingly from this patent application, if you look at figures 1C and 3B up at the top, you can see that compound 31 elicits dose proportional reduction in tumor volumes in this xenograft model. And that reduction, that activity is actually sustained after termination of dosing, as you can see in figure 3B, for several days afterwards. So this is a really exciting program that looks like it holds a lot of promise in a fairly difficult but exciting and emerging area of drug discovery. So the next program I'll talk about, this kind of falls under the umbrella of a really important area of research. So almost any key advancement in this area is pretty exciting. And so this is Blacksmith's Therapeutics LpxC inhibitors for gram-negative bacterial infections. And as we all know, we need more antibacterial medication. So it's exciting to see some progress in this area. So this patent application actually disclosed prodrugs of compound A. So compound 1 on the left is a phosphate prodrug of the active moiety compound A on the right. So what is LpxC? It's a lipopolysaccharide biosynthetic enzyme that's really key to the survival of the bacterium. And what Blacksmith Medicines has disclosed previously is the clinical or preclinical efficacy data for its lead compound, FG960. This is a compound that they got as part of the Forge Therapeutics efforts. However, they have not yet revealed the structure of FG960. But what they've revealed with FG960 is that it shows strong activity against multi-drug resistant Enterobacterales. So that's really important activity in a really tough bacterium. Furthermore, FG 960 is advancing through IND enabling studies. So it's really at the threshold of entering the clinic, and that's really exciting. Blacksmith Therapeutics also has another compound that's approaching IND enabling studies, FG2101. And so one thing that's exciting about these two compounds is that they're both non-hydroxamates. And these compounds, specifically 960, has shown great antibacterial activity, but it has not displayed the cardiovascular toxicity seen in those earlier hydroxamate-based inhibitors. So if we look at tables 2 and 3, you can see that regardless of whether you dose IV or PO, the compound 1 really leads to good exposures of compound A. And you also can see that there's a longer half-life in dogs versus rats. And then finally, in table 4, in mice, compound 1 and compound A both significantly reduce bacterial loads compared to vehicle control in a dose proportional fashion, particularly if you look in the kidney. So just like with the previous program, if you look at this data and these compounds are getting away from that problematic structural motif that's been a part of other previous antibacterials in this space, this looks like a really exciting program, and it holds a lot of promise in a difficult area. So we're going to be watching, this program and related programs very closely at Drug Hunter. And with that, I'll hand it off to, I believe, Dennis again, doing double.
Yeah, that's right.
duty.
Yeah. Thank you, Lew. Yeah, no, that's super exciting. So 2 more targets that we want to cover here today. And yeah, the first one, of course, needs very little introduction to this audience here, is the GLP-1 receptor. So the GLP-1 receptor, as we know, there are a lot of, there's Ozempic, of course, those peptides are approved drugs, and they've also been recently made orally bioavailable, right, with like some formulation tricks that we have learned. But, there was a really interesting presentation at the EFMC in Rome that we attended that said that there's not enough starting material on the planet to supply the incredibly high demand for these types of peptides. And we've seen shortages of those peptides. We've seen shortage of tirzepatide, which is Eli Lilly's Mounjaro. So there is definitely a need for small molecule alternatives. And, of course, we could also think about increasing the oral bioavailability here. Recently, so of course you're all aware of danuglipron, which is Pfizer's GLP-1 receptor agonist, and then orforglipron, which is also the most advanced small molecule GLP-1 receptor agonist, which was discovered by Chugai, but now acquired by Eli Lilly. And recently, at Drug Hunter, we've been scrubbing the INN list, which is really interesting, and I encourage you to take a look at this article. And we found out that Structure Therapeutic named one of their GLP-1 receptor agonists eleniglipron. And we wrote an article about this, and it's a really interesting phosphine oxide structure that basically we believe is the identity of their clinical candidate. But yeah, so really, of course, there's been a lot of activity going on in that space, and GLP-1, the GLP-1 receptor, of course, as you all know, is a class B GPCR, and class B GPCRs are really actually beasts to drug, right? So you can see that by the monsters that emerge against this particular target class. So if we think about the natural peptide GLP-1, that of course exhibits anti-diabetic incretin effects and increases downstream the cAMP levels and thereby of course the downstream signaling. So oftentimes though that agonism of the GLP-1 receptor can also result in recruitment of beta-arrestin, and that then in turn leads to internalization of this GLP-1 receptor. And of course, this is then followed by desensitization. So, we want to make sure that there's a differentiated profile with respect to this recruitment of beta-arrestin. And actually, danuglipron shows detectable recruitment of this beta-arrestin, whereas orforglipron doesn't. And then a few other differences. Orforglipron is also a partial agonist, whereas danuglipron is a full agonist with respect to the CAMP activation. And still the minute details in the clinic have to be kind of worked out how those things differentiate. But yeah, so GLP-1, of course, super hot. And we've monitored the space, of course, and we've seen the entry of Gilead to this space, which is kind of surprising since they're typically not in metabolic diseases, at least not known for that. But they entered the space in actually 2021 when they were interested in an agreement with Novo Nordisk using their semaglutide, better known as Ozempic, in a fixed combo for the treatment of NASH. So, liver disease, non-alcoholic liver disease, right? So they were interested in using their own FXR inhibitor, cilofexor, and their own ACC inhibitor, Fircostat. But now, apparently, they're interested also in coming up with their own GLP-1 receptor agonist, small molecule. And we've been watching this space, and they've made some interesting changes here to danuglipron, which really led to a dramatic improvement in its potency, in the on-target potency. You can see orders of magnitude more potency here, and also, importantly, oral bioavailability, which is a problem with danuglipron, only 3% in monkey. Has gone up with this derivative here that Gilead disclosed. I mean, Gilead actually has a clinical candidate right now in that space. They still stick to the line that they are working on it for NASH. The clinical candidate is called GS 4571, so watch out for this. Gilead actually recently reported data in an obese monkey model where they showed that a decrease in body weight of 6.5% over 36 days only. That really drove the stock price of Gilead up, as you can think, but still Gilead is sticking to the line that they're interested in this for NASH. But of course, it could be a better version of danuglipron, just comparing the data. Novartis is also in this space as well, and also has released a patent also earlier this year on a similar, on the same target. And, they actually have shown some differentiation in terms of data from danuglipron in that, this compound that they're patented here has some differentiation with respect to less beta-arrestin recruitment and less receptor internalization compared to danuglipron, which is great. The last target that I want to cover, and the last kind of patent highlight that we want to cover before we launch into the questions, is this really somewhat revolutionary patent, I think, that Monte Rosa has filed. And Monte Rosa also recently published, I should say, a bioarchives presentation on this. So why is this so interesting? Actually, if you went to the EFMC, and we have some coverage on the website, Magnus Walter gave a phenomenal presentation about this at the EFMC in Rome. And really, this work suggests that you can teach the old horse, if you will, Serblon, some new tricks with respect to degradation. So molecular glue degraders, of course, are a really, really hot field, an emergent class, of small molecule drugs that induce proximity between ubiquitin ligases and target proteins that cause their degradation through the cellular waste disposal system. I mean, most prominently, thalidomide and its analogs have shown clinical effects binding Serblon, which, of course, the ubiquitin ligase, right? And then rewire the substrate selectivity, basically creating a Serblon drug neosurface that is competent in engaging so-called neosubstrates and neosubstrate proteins, I should say. And those neosubstrate proteins, most famously, probably IKZF1 and IKZF2, but also WIZ and GSPT1, they can then essentially be degraded through this type of mechanism. So really, really interesting. And by looking at those types of, client proteins, if you will, a common structural surface area has been found that's really, you know, similar to all in all of these peptides, or in all of these, you know, yeah, peptides, I should say. So basically, if you look at them, it's a G loop. It's a so-called G loop. But really, Monte Rosa has gone the extra mile to identify targets that maybe don't feature this particular G loop. So they have like this really cool surface screen, and you can read the details about this in their presentation. Matt on our team said that that should be required reading for anybody who works in the field of molecular.
I mean, really, yeah, I think revolutionary is quite right about it. I mean, you know, one of the things that they really did in this bioarchive publication was show that you don't need to rely on the primary or secondary structure of these proteins to be able to predict if they're going to be substrates of cereblon. It's about the surface homology. You know, VAV1, you know, the G loop is making sure it has that glycine that's really keen in engaging cereblon. The RT loop that VAV1 has doesn't have a glycine in it, and it's able to engage cereblon. And I just love this, that we've reproduced a couple of the figures here. The volcano plot on the top left is honestly one of the cleanest volcano plots I've ever seen in my life. I've seen a lot of chemoproteomics work. So yeah, it's really, really exciting and fun to watch. And if anyone is interested in glue degraders, really should read this paper that's out.
Absolutely. And yeah, VAV1, as you said, Matt, is a protein that has been traditionally hard to drug. There's really no, you know, druggable, you know, volume and close volume. It's really difficult. There have been attempts, I think, at allosteric VAV inhibition, but so far none of the compounds made it to the clinic. Of course, Monte Rosa actually has a clinical candidate in phase 1. That's called MRT 6160. And yeah, VAV1 is just really this very interesting protein that is expressed, I think, in blood cells, and, you know, mainly T cells and B cells. And, you know, those structures that you see here, we highlighted in our patent highlight. And, you know, after that patent highlight published, the Monte Rosa bioarchive publication came out. And as you can see, you know, it's a very similar looking structure here, compound 6 that they detailed. And, you know, as Matt said, the volcano plot is really impressive, but they were also able to show really in really interesting models here for encephalitis, for example, superior results to dexamethasone, which is super, super fascinating. Their platform is actually accessed by other large pharma companies such as Roche. So Roche actually paid $50 million to have access to Monte Rosa's platform, and they're basically in a collaboration program here. The platform is called Queen. So yeah, we believe that this is a target definitely to watch out for. And again, I think changes the paradigm of how we should think about molecular glue degraders and how to maybe really, you know, be organized better in a way, for a lack of saying this in any better way, discovering those molecular glues. So I think Monte Rosa really showed that here, how to do this. Yeah, and with that, if you
guys, I'll just chime in as somebody who's worked in this space.
Yeah,
I can assure you there are a lot of companies out there that are expanding the scope of you know, proteins that can be degraded by cereblon, and the variety of Degron motifs is likely going to be exploding in the next 2 to 3 years. But people aren't just in a position to publish a lot of this stuff at this time.
Totally. Yeah. Yeah. Thanks for your perspective, Lew. I yeah, totally believe that the field of
I wish I could say more, but I can't.
To be disclosed. To be disclosed. Yeah, and with that, really thank you. I mean, you can find all of these case studies on Drug Hunter as well, and hundreds of more things. So, you know, give us a visit. And really, you know, Lew, Matt, and I, we get to talk about this, but it's really a team effort putting this together. So we are basically consumers of our own product. So I read Drug Hunter every day to stay on top of what's going on. So really thank you to the team for putting this really great resource together. And now we can launch into questions. Thank you for listening to us, and we'd be excited to answer your questions. And with that, I'll check if there are any. Lew and Matt, if you can also help prioritizing questions if you'd like. All right, I see one question by Gagan already. Would the target search fetch the target name anywhere if it appears in the patent? If that's the case, there would be a lot of noise. Okay, so
no. So this is one of the real strengths of kind of our patent database. I mean, obviously you can use some of the competitors and they'll give you 10,000 patents and you search for KRAS. But really what the Drug Hunter platform does is we have these annotations, which are, these are not AI generated either. These are from industry scientists looking through it and picking out the key points people would be interested from. And it text searches that rather than the whole patent, right? There are other tools out there if you want to crawl through the entire patent database. But like you said, you're going to get a lot of noise, whereas a Drug Hunter will really help you cut through that noise through these annotations. So it's text searching that.
Probably makes sense. Yeah, thank you for the answer, Matt. And we have another question here from Greg Mignus. Is patent searchability only for premium membership or individuals? So yeah, currently the patent search tools, all the tools that we showed today are available for our members and are not open access at this moment.
But really, I think we've been able to show today that the value that these tools really have, and be able to pick out these trends and like Dennis said, really predict actually what some of the big moves are in the M&A space, right?
Totally, yeah, and we're seeing more and more investors actually coming on board at Drug Hunter, so that might be an indication for the growing value that I think we bring to the community. All right. Are there any more questions? Yeah, audience, feel free to populate the Q&A box. You can also, at this time, I think, use the chat. We will be able to prioritize questions from the chat. Yeah, if there's not anything more, we can also happily close it here. Matt and Lew, any comments that you wanna make before we part?
No, just echo what you said, that I consume my own product, right? It's there's so much out there in drugs, Drug Hunter really does. I obviously write some of the articles and edit some of them, but I'm always happy to see contributions from our other team members, because I know they're going to do a great job in being able to teach me what's going on outside of what I'm covering.
Yeah, and I'll just say, having worked at Drug Hunter now for 8 months, I'm pretty humbled by how much incredible drug discovery research is going out there in the world. And, I've come to appreciate even more the value of Drug Hunter and sort of, monitoring what's going on and really identifying the most important things and then bringing that to the attention of people. So, yeah, if you're not a member of Drug Hunter, I highly recommend it. You will learn a lot.
Yep. Definitely agree. Yeah, it's yeah. I mean, I have to say, like, my breadth of knowledge exploded since starting at Drug Hunter, right? Like, as oftentimes in the industry, you're really focused on one target, but Drug Hunter has, I think, so much to offer to widen your horizons on other targets, and so many times, really, there some learnings that you can actually transfer from other projects that are not necessarily your target that you would have otherwise missed, that you can, pick up from just, 10, 15 minutes of reading Drug Hunter every day. Yeah, we do have a question here from Ann Tran, and she's asking the INN list, how often is it updated? So, Ann, the INN list publishes every year, twice in a year, I should say, the proposed list. And yeah, we have coverage of that. Like, from now going forward, we're intending to make those lists searchable, so that you can find these targets, that you can find these interesting structural motifs faster than you would. So we're really trying to build this for the community. And I think that's another great example of a feature that was actually suggested to us by the community. So multiple people actually approached us with the request to add this INN list. One of them was the CSO at I think Alamis. So yeah, we really try to implement all of the things that we think would be useful for any drug hunter out there. And if we hear suggestions multiple times, I think that indicates to us that this is important to you. So yeah, thanks for the question, Anne. Yeah. So I think with that, if there are no more questions, thank you, Matt. Thank you, Lew, for weighing in today with your expertise. Really enjoyed this. I, again, learned a lot from you guys, as I do every day. Thanks to our audience also for dialing